Good morning and good afternoon. Welcome to this webinar, Accelerating COVID-19 Drug Discovery, Qualified Cell-Based Assays for Pro-Inflammatory Cytokines, brought to you by Xconomy and presented by Eurofins DiscoverX. My name is Brianna Kossifret with Xconomy. Before we begin, please let me explain how you can participate in today's session. First of all, if you have any technical difficulties during today's webcast, please press the Help button on your player console to receive assistance in solving common issues. We welcome your questions during today's event. In order to submit your questions to today's presenter, simply type your question into the Q&A window on the left-hand side of your screen, and then make sure you hit that Submit button. We'll be answering as many questions as possible during today's Q&A that will follow the main presentation, but please feel free to send in your questions at any time and we'll add them to the queue. If you are having audio difficulties or trouble advancing the slides, simply hit your F5 to refresh your webcast console. And lastly, our last announcement today, please make sure you're aware that today's session is being recorded and will be available on the Xconomy website, xconomy.com, beginning tomorrow for you to review. You'll be notified by email when the archive is available. And now let's introduce today's presenter. We have Gaurav Agrawal, PhD. He's a Scientific Development Manager for the Marketing Department at Eurofins DiscoverX. Gaurav, the floor is yours. Welcome.
Thank you, Brianna, and hello everyone, and welcome to this presentation. I'll briefly introduce myself. I'm Gaurav Agrawal, Scientific Development Manager at Eurofins DiscoverX. My role here is to support scientific integration of our cell-based assay platform in drug discovery programs globally, and thereby building strong partnerships with biopharma industry. So thank you for joining me today. The webinar here is mainly focused on how our qualified cell-based assays can accelerate the development of therapeutics targeting pro-inflammatory cytokines that are implicated in COVID-19. With over 40 million cases and over 1 million deaths, the fast-moving pace of COVID-19 pandemic has been challenging the capacities of healthcare systems worldwide. As we have seen the pathology of COVID-19 in severe cases, patient's immune system mounts a highly disproportionate and aggressive inflammatory immune response. For that reason, there are several ongoing clinical programs that are evaluating therapeutics targeting these inflammatory signals as a treatment strategy for COVID-19. Now, the high mortality rate in COVID-19 patients has been linked to cytokine release syndrome, commonly referred to as cytokine storm, which is an excessive and dysregulated release of pro-inflammatory cytokines by body's immune system. This leads to an uncontrolled inflammation leading to acute respiratory distress syndrome, or ARDS. This is evident from highly elevated levels of cytokines in patients with ARDS. Here I have put together a quick recap to pro-inflammatory cytokine pathogenesis in COVID-19. The initial point of contact with SARS-CoV-2, as we know, is the ACE2 receptor on the alveolar cells. Once the virus infects the alveolar cells, they trigger an innate immune response in the host, where the immune cells in the lungs releases inflammatory signals like interleukin-6 and other chemokines to attract other immune cells to the infected tissue. Now from here, we can go two ways. First and most common, where things go as expected and the immune response mounted is proportional. The infected cells are cleared by the CD8 positive T cells and macrophages, followed by B cell activation, producing antibodies, which helps in clearing the virus. But it can very well go in a different direction, where the body mounts a dysfunctional immune response to the infection. This is mainly caused by cytokine storm, where pro-inflammatory cytokines such as interleukin-6, interleukin-1 beta, TNF-alpha, GM-CSF are excessively released by the immune cells in the lung and its associated tissue. In fact, the level of these cytokines are highly predominant in COVID-19 patients who are in ICU. These cytokines cause acute inflammation of lungs and can lead to death due to ARDS in severe cases. Now, it is still an active area of investigation as to what factors makes the immune response go one way or another. But in broad terms, these factors are associated with age, smoking status, or pre-existing conditions like heart, lung, or kidney disease. Diabetes or cancer may play an important role in disease progression. But cytokine storm remains one of the main reasons behind the severe implications of disease, and even when the patients survive, there are long-lasting implications which are mainly associated with tissue injury caused by immune cells. The patients will be prone to getting secondary infections and multiple organ failures, which may lead to death. So this is very serious in COVID-19 patients and has long-lasting effects. So what are we doing about combating cytokine storm? There are hundreds of active clinical trials ongoing to test both small and large molecule therapeutics for the treatment of COVID-19. As expected, most of these trials are focusing on repurposing the drugs that are already approved for other conditions. Here I have listed the cytokines and chemokines that are mainly implicated in cytokine storm with their respective therapeutic drugs that are currently in clinical trials, such as Tosylusimab targeting interleukin-6, anakinra targeting interleukin-1, adalimumab neutralizing TNF-alpha, and each of these drugs are a part of several clinical trials across the globe. Now, the figure mostly lists repurposed drugs, but there are also many new therapeutics such as GM-CSF and IL-8. Now, for development and characterization of these therapeutics, as well as releasing them into the clinic, reliable functional assays are required. For this, this is where Eurofins DiscoverX can partner with you with our reliable cell-based assay portfolio. We have functional cell-based assays for each of these cytokines and chemokines. And most importantly, we also have qualified and MOA-based bioassays for all these cytokines as well. These bioassays can really accelerate your drug development and release programs, and how they can do that would be the topic of my rest of the talk. So in this part of presentation, I will introduce you to our cell-based assay platform and present a case study for Tosylusimab bioassay, which is designed for potency and stability testing for therapeutics targeting interleukin-6. With that, I will give a quick overview of the bioassays with three other relevant targets. As you see, these bioassays have been qualified with marketed drug, which makes them highly relevant for biosimilar and biobetter drugs. Following this, I will discuss our signaling reporter assays, which provide a more downstream readout of these pro-inflammatory cytokines. And together, these assays can provide a comprehensive data for characterizing therapeutic drugs targeting these cytokines. And towards the end, I will tie it with another set of assays for characterizing small molecule drug candidates and discuss functional data for evaluating JAK1, 2, and 3 inhibitors, which are actively investigated as a treatment strategy for COVID-19. But let me first briefly introduce our enzyme fragment complementation, or EFC, which is our core technology behind these assays. So our assays utilizes this proprietary technology, which is based on split beta-galactosidase system, where the enzyme, the beta-gal enzyme, is split into two inactive fragments called ED, or enzyme donor, and the EA, or the enzyme acceptor. When these two fragments are put together, they complement each other to form a functional beta-gal enzyme. We add a chemiluminescent substrate in the assay, and with that, the active functional enzyme hydrolyzes to produce a light signal that can be read with any benchtop luminometer found in most labs. As an example, here is an assay outline for the fibroblast growth factor receptor, or FGFR. So the natural MOA for FGF receptor, after binding to its agonist FGF, the first significant event that happens is the recruitment of a SH2 domain protein to its intercellular region. That's exactly what we have used as an MOA here using the EFC technology. The FGF receptor is tagged with one of the complementing beta-gal fragment shown in red here, while the SH2 domain containing protein, the SHIP protein, is tagged with the other complementing beta-gal fragment, the EA. Now, when the FGF ligand is added to this assay, the receptor gets activated, leading to the recruitment of the EA-tagged SHIP protein to the intercellular tail of FGFR, and thereby complementing the inactive beta-gal fragments and forming an active beta-gal enzyme. Next, the cells are lysed and the detection reagent is added, which contains the lysis buffer and a chemiluminescent substrate for the beta-gal enzyme. Now let's look at another example where we have used the EFC technology for a receptor dimerization assay. Here is an example for interleukin-1 receptor and its co-receptor IL-1 associated protein. These receptor subunits dimerizes in presence of interleukin and represents the physiological MOA of interleukin-1 action. As you see, these receptor units are tagged with beta-gal fragments, and as I described in the previous slide. Now, in presence of interleukin-1, the receptor subunits dimerizes, thus bridging together the beta-gal fragments. The EFC assays are highly quantitative, as you will see in the later slides, typically producing a gain-of-function response. Similarly, the assays can be used to quantify inhibitory signal from the same assays. As in this case, I have shown an example with anakinra, which is an IL-1 antagonist protein. In response, our EFC assays produces a quantitative signal, quantitative inhibitory signal in a dose-dependent manner, as you can see here. Both the agonist and the antagonist assays are very specific and produce a very nice, robust assay windows. And likewise, we have successfully utilized EFC technology for many other assay formats, such as to quantitate protein-protein interactions, protein turnover and degradation, for evaluating downstream responses and protein secretion or membrane translocation. Also developed are several cell-based assays for intercellular trafficking and internalization, as well as biochemical assays for compound target engagement, and more recently for developing cytotoxicity assays such as ADCC and ADCP. For our cell-based assays, we do offer phase-appropriate assay formats. One is, of course, the stable cell lines, offering a continuous culture format. But more importantly, we have bioassays, which are ready-to-use format and don't require any cell culture. It is our highly optimized assay format developed for potency and stability testing and are perfect for implementing in QC lot release programs. We also took a step forward and qualified our assays with marketed drugs where they are available. Now, before I move forward, let me give you a very quick overview of a general assay protocol for our cell-based assays. The protocol more or less remains similar across our EFC platform, and for this reason, it is easy to perfect these assays quickly in a lab, as well as transfer and implement these assays at multiple sites. Now the assay starts with plating cells in assay plate, usually overnight. Next day, the molecule of interest is added and incubated for another 1 to 3 hours. Thereafter, you will add a detection reagent and the beta-gal substrate and incubate the plate in dark for an hour to 3 hours. And this is about it. After the incubation, the plate can be read in any benchtop luminometer. The schematic on the lower right side shows a bioassay kit that comes with all necessary reagents, tried and tested, the certificate of analysis document that comes with each kit. So with these bioassay kits, there is no need to source any of the reagents from any external vendor. That keeps the variability in the assay to a minimum. Perfect. I think we are ready to review a case study for one of our qualified bioassay. I've selected our Tosylusimab bioassay since it is for interleukin-6 targeting therapeutics, which is the most predominant in the cytokine storm syndrome, and therefore very relevant to COVID-19. The intent of this case study is to provide you with an overview on how we go about qualifying our bioassays to make them most suitable for potency and stability testing for drug candidates when implemented in lot release programs. So let's look at the assay design for this bioassay. And of course, it is based on our EFC technology and utilizes a dimerization platform, very much like the interleukin-1 assay that I described above. Now, in presence of interleukin-6, the receptor units dimerize, bringing the receptor units together and thereby complementing the EFC fragments. Adding the beta-gal substrate produces a quantitative signal. However, when the assay is run in presence of Tosylusimab, an anti-IL-6 receptor antibody, the binding of interleukin-6 is inhibited in a dose-dependent manner, as you will see in the schematic figure here, and produces a quantitative inhibition curve. Now, here are some of the key assay parameters we assess during assay qualification. Firstly, we assess assay consistency between several dose response curves. This gives a very good idea on any variability associated with the assay. Second, we do look at plate uniformity across the entire plate. This provides us with any plate associated bias and possible edge effects. Next, we also look at assay repeatability and for consistency in slope of the curve. And lastly, perform assay suitability and relative potency analysis. We determine assay accuracy, precision, and linearity over a range of 50 to 150%. And this is performed by two individual operators. Here is the data for testing intraday plate-to-plate variability. In this slide, you see 3 experimental replicates of Tosylusimab assay that were prepared as 11-point dose response curves and run in the same plate. Each plate has duplicate pills per dose on the same day by one operator. Similar experiments were repeated again to obtain data from 3 individual days for inter-day variability. As you can see, the curves are very uniform in these experiments, and in the next slide, we analyze the data statistically for intraday and interday variability. The table on the left shows analysis of each curve with excellent interplate repeatability and intermediate precision of both assay window as well as the IC50 values. The precision was within 8% for signal to background and below 10% for IC50s. This is an excellent data for experiments run on the same day in separate plates. Now the table on the right shows a culmination of 3 such experiments to calculate inter-day precision. It is below 25%, as you can see, which is again very well within the acceptability criteria, which is set at 30%. In all these data sets, they are suggesting an excellent precision between multiple sets of experiments run across multiple days, and this is something that's required when the assay is implemented as a in a quality control environment for drug release. Okay. Next, we perform relative potency experiments and parallel line analysis with Tosylusimab across a range of 50 to 150%. The experiment here shows data from 2 individual analysts. The blue curve is with 100% reference, while the green is the sample. As we move from 50% to 150%, the green curve shifts towards the left and moves across the 100% reference, depicting an increase in potency. PLA software was used here for analysis, but we can also do this analysis using a Softmax Pro software as well. Here's a statistical analysis of the relative potency data I showed you. The assay was determined to be very accurate with assay precision of 105% and an overall assay inaccuracy with a 9.3%. A high degree of assay accuracy here suggests that the bioassay can measure relative potency with high confidence. On the other hand, assay precision was below 6%. Such high degree of precision here shows an excellent assay repeatability when performed on different days and between different analysts. Dilutional linearity was analyzed, plotting the measured against the expected relative potencies as shown on the table on the right. The R-square value came just below 99.3%, showing an exceptional assay linearity. These datasets that I discussed with Tosylusimab bioassay here is a part of our standard assay qualification work for our qualified bioassays. With such amount of assay qualification work behind them, you can be confident that when implementing these assays in a quality control space for your biologic or biosimilar drug candidate release, these assays are going to produce very uniform data across the life cycle of the drug. Next, I will discuss 3 other bioassays briefly and not go over the entire qualification work as I shown here, but please feel free to reach out if you would like to review them. Okay, so the next bioassay that I will discuss is for anakinra. I did went over the working of this bioassay in the beginning, but to quickly recapitulate, the bioassay design here, like all of our bioassays, is MOA based and MOA reflective. The assay is used to quantify an inhibitory signal from anakinra, which is highly modified form. This slightly modified form of the naturally occurring interleukin-1 receptor inhibitor. As shown in the bottom panel here, in presence of anakinra, there is a dose-dependent inhibition of interleukin-1 receptor dimerization. So let's look at the functional data. Here in blue, the blue curve, the dose-response curve, is for interleukin-1 beta, with the purple for anakinra. The assay produces a robust assay window with sensitive detection and a dose-dependent response to anakinra. We further performed assay qualification work as I've shown for Tosylusimab bioassay, and here the data shows dilution linearity derived from relative potency experiments performed over a range of 50 to 150%. And as you can see, the relative potency data derived from this bioassay was highly accurate and with over 99.4% recovery, and it's also very precise. The next bioassay I would like to highlight is for GM-CSF, which was qualified using the marketed drug Leukin. This is a recombinant sarcoma stem produced in yeast. Both sarcoma stem and inhibitory antibodies against CSF2R receptor, such as Linzolumab, are in clinical trials for COVID-19. Similar to Tosylusimab and anakinra, this bioassay is also based on receptor dimerization. So let's quickly look at the qualification data here with Leukin. As you can see, this is one of our best performing bioassay with dilution linearity over 99.5%. Also, the relative potency data derived from this bioassay was highly accurate and also shows a very high precision as well. Now, the last bioassay we'll review would be the Adalimumab bioassay targeting TNF-alpha. As we know, TNF-alpha signaling goes through NF-kappa-B pathway. The TNF-induced phosphorylation of I-kappa-B makes it a target for proteasomal degradation. And so in this assay, we tag the I-kappa-B protein, as you can see, with the EFC tag. The levels of the I-kappa-B protein is inversely proportional to the TNF-alpha concentration, and thus it forms a quantitative loss of signal assay. In presence of TNF-alpha blocking antibody adalimumab, the I-kappa-B protein is not phosphorylated or degraded, thus restoring the I-kappa-B levels in the cell, and hence produces a gain of function response with the inhibitor. Here is the functional data. The curve in blue, the dose response curve, is for TNF-alpha, and purple for Humira. The assay produces a robust window with sensitive detection and dose-dependent response to both TNF-alpha and Humira. The assay qualification for Humira also aligns very well, producing, I will say, an almost perfect dilutional linearity with 99.99% and with excellent accuracy and precision. Okay, now let's put things in perspective here. What you see here is a typical roadmap for developing a fit-for-purpose bioassay for a biologic drug candidate. which is indeed, true for most of the drugs released into the market for clinical use. It can take upwards of 12 months if starting from cell line development, subsequent qualification, and at the end, implementation for QC lot release process. Now, this time frame, what you see here, can be significantly reduced by more than 9 months when you implement one of our qualified bioassay. Here you can see right away the assay can be transferred to a CRO site where they can start the assay validation work right away. Now this way, our bioassays significantly accelerate time to market for therapeutic drug candidates and help save significant time and cost as well as resources for producing these assays in your own labs. Next, I would like to introduce our new set of reporter assays designed to provide a more downstream readout for activation or inhibition of a signaling pathway. These would be highly complementary to our bioassays that are more focused on recording upstream events. In the schematic here, a signaling event induced by a ligand binding jumpstarts a transcription event for a reporter protein that is tagged with our EFC tag. Now this EFC tag can be detected by the addition of other complementary EA protein, thus forming a functional beta-gal enzyme. And then we can add the beta-gal substrate, and the assay produces a highly quantitative and gain of function readout. I will show you now 3 different reporter assays that we have built using this technology for cytokines that are relevant to COVID-19. The first is interleukin-6 itself. The very eye of the storm, I will say, is interleukin-6 for cytokine storm in COVID-19 patients. The assay is built in HepG2 cell background, exploiting the STAT3 pathway, whose role is well documented for cytokine signaling. The assay was used to analyze antagonist activity of Tosylusimab. We saw a robust and sensitive response to interleukin-6 and a good inhibition curve for Tosylusimab with a large signal-to-background assay window for both. Next in our NF-kappa-B reporter assay is the U2S in U2S cells for TNF-alpha. Now TNF-alpha, like many other of its family members, signals through NF-kappa-B pathway. The assay was used to analyze antagonist activity of Humira. Now we saw a robust and sensitive response to TNF-alpha and a good inhibition curve by Humira with a large assay window over 50-fold, as you can see here. The third assay I would like to mention here is for RANK. RANK is a member of TNF-alpha family and binds to RANK ligand. The reporter assay overexpresses the RANK receptor in a U2S background, also utilizing the NF-kappa-B pathway as a signaling event. The figure here shows a highly sensitive RANK dose response curve with over 20-fold assay window, and on the right, a good inhibition curve for Prolia as marketed for denosumab, which is an anti-RANK antibody. Thus combined, these assays, these bioassays and signaling reporter assays will provide a comprehensive understanding for drug molecules MOA. Now in this last section, I would also like to highlight our functional cell-based assays for small molecule inhibitors. In particular, would discuss our assays for JAK inhibitors. In the past 6 months, JAK inhibitors have become an active area of investigation for several ongoing clinical trials evaluating the therapeutic potential of JAK inhibitors for the treatment of COVID-19. Among them are ruxolitinib and baricitinib are the most prominent ones. Now, these inhibitors can have a distinct advantage over other immunomodulatory strategies in COVID-19 as they can exert a dual effect. Firstly, an anti-inflammatory effect to block multiple pro-inflammatory cytokine signals simultaneously, as well as antiviral effects by impeding viral endocytosis. So by these 2 mechanisms, these inhibitors are much preferred route as a treatment strategy for COVID-19. Now, the data on the right here is a functional assay where JAK1 is expressed as a cytosolic tyrosine kinase along with CSF3 receptor. Now, when GCSF is added, which is the ligand, CSF3 receptor is activated and is phosphorylated by JAK1. Similarly, the assay in the middle expresses TEC2 with growth hormone receptor and JAK3 with prolactin receptor in the JAK3 activity assay on the left. Now, together in these assays, we have tested several JAK inhibitors. Most relevant among them are shown here, such as ruxolitinib itself, which is a JAK2 inhibitor. There are other inhibitors, as you can see, for JAK3 assays as well as JAK1 assays as well. So taken together, these assays gives you a big repertoire of cell-based assays for classifying these JAK inhibitors, which are relevant for COVID-19 therapeutic strategy. I think I'll stop here and would like to quickly mention that we have a strong custom assay development team who can build assays or modify existing ones to suit your needs. We can also take up pilot studies if you would like to run quickly some feasibility study and other assays. We also have Eurofins Discovery Partner Labs who can run these assays as a full service for you. Now, quick set of conclusions here. In this talk, we have discussed our target-specific qualified MOA reflective functional assays for drugs targeting pro-inflammatory cytokines, all of which are implicated in COVID-19 pathology. With that, we offer multiple solutions, including custom assay development service. One relevant example is our ongoing custom assay development project for ACE2 receptor that would be intended for characterizing therapeutics aimed at preventing the coronavirus from attaching to the alveolar cells in the lungs. And for more information on our assays, please visit our website at discoverx.com. With that, I would like to thank you for joining me today. I will be happy to answer any questions now.
Gaurav, thank you very much for an excellent presentation. Before we get going into our Q&A session, I just want to remind you that how to ask a question. Make sure you submit your questions now in the Q&A widget on the left-hand side of your screen, and then make sure you hit that submit button. A few of you have already submitted some questions, so Gaurav, why don't we go ahead and get started with the Q&A, okay? Here's our first question today. What are some advantages to implementing your bioassays versus using a cell line in GMP lot release programs for drug candidates?
Thanks, Brianna. Yeah, that's a really good question, and of course require a detailed answer. But I'll quickly mention a few things here. The cell line versus the bioassay has been, kind of the whole idea on us developing the bioassay here. So there are several advantages. Bioassays gives a much better reproducibility, robustness, and day-to-day precision that you are expecting when you are implementing these assays and lot release, as I've been mentioning during the course of the talk. Cell lines, of course, are implementable, but requires more work. Will produce, I will say, relatively variable data compared to bioassays because there are certain things we do to optimize bioassays for them to produce a very consistent data day to day. So those are very distinct advantages with bioassays. Cells are frozen at the same time, so you have very synchronized cells that you are using in the assay versus the cell line, where you will see passage-to-passage variability in there. So yeah, there are several distinct advantages of using the bioassay versus the cell line. Plus, there are, of course, other advantages as well. Our bioassays can be transferred to a CRO site, and there is no transfer fee to that. So, including those, we definitely envision the bioassays will be our preferred format for transferring to a third-party site for easy implementation and a high success when you are implementing a QC lot release-like program.
Great, thank you. All right, here's our next question. How do you ensure an uninterrupted long-term supply for your bioassays in order to support drug release programs that may run anywhere from 10 to 20 years?
Yep, that's a great question, and we completely understand these are some realistic things that everybody has to kind of take into account when they are transferring an assay or implementing an assay in such a long-lasting program such as a QC lot release. We do have process in place where we can ensure a long-term supply. We do have processes in place, as I said, to put the paperwork where you can be assured that we will be there to support your program for the life cycle of the drug. So those things we definitely ensure that we put in place before we put the assays in a QC lot release program. So we'll work with you and make sure that you are well supported, your program is well supported. And we have been doing it for multiple years, for multiple drug discovery programs. So pretty much we have it covered at our end. So that's kind of a peace of mind for you that when you're implementing the assays, everything is there that we can ensure a long-term supply for reagents, cells, and everything.
Okay, perfect. All right, our next question here, Gaurav. Do you provide these assays as a service if we would like to run a short feasibility study with our drug molecule?
Yeah, excellent question. Yes, exactly. We do that. And that's kind of a very important point as well, because it's important sometimes to ensure the drug molecule is working in the assay. Of course, there are so many innovative ways people are manufacturing these drugs. They're attaching, receptor subunits, different moieties, glycosylations, things like that. So they are definitely altered molecules to make them better. So it's an important step that people need to ensure that these molecules are responsive in the assays, they are functional. And for that, our custom assay development team provides a service where we can run these assays with your drug molecules and get basically a pilot study where you will see a data set that basically shows that the assays are working, what the reproducibility is. And we are here to help as much as we can. So we can also run a systematic qualification study as well, where we will look at precision, accuracy, linearity, parallelism, and things like that to show that the drug molecule is basically very fit to be used in these assays before you're transferring to a CRO site. And that makes the assay transfer very easy as well. So yeah, we do provide services at different tiers. As I said, we are here to help as much or as little as needed here. And we have these services that we can provide to make your decision, basically de-risk your programs, make your decisions more and more confident using our assays.
Perfect. Thank you, Gaurav. Our next question here. How are the EA/ED added to the cells?
Yeah, great question. So yes, we have, as one of the slides showed, we have a lot of different formats for these assays, and these formats, some of them expresses the EA and ED tags that are already tagged with the receptor subunits and expressed in the cells. So in this case, when the biological event is happening, as for example, the ligand is being added to the cells, your receptor fragments come together, or your protein is recruited, as in case of the SHIP protein example I showed. So either these EA and ED fragments are already expressed in the cells, tagged to the respective receptors or the recruiting protein, or in some assay formats, such as our cytotoxicity assays, one of the fragments is expressed along with the receptor in the cell, while the other fragment has been added alongside the detection reagent. So it definitely depends on what assay format we are using. There are so many intelligent ways we have designed our assays so that ED and EA subunits either can be expressed together in the cell, or we can bring in the EA to make the assays more specific. So yeah, there are different ways we can use these fragments to build different assay formats.
All right, Gaurav, let's see here. We have one more question here. As a reminder, if anyone needs to submit a question, please make sure you do so now. The Q&A widget is on the left-hand side of your screen, and then make sure you go ahead and click that submit button so we can get your question into the queue. All right, another question here. Have you tested a variety of cell lines originating from different organs? And if so, do the results vary?
I think it's a great question, and of course, very relevant one as well. We have a very, I'll say, extensive experience working with many different cell backgrounds. I believe we have over 45 different cell backgrounds. The assay is built on 45 different cell backgrounds. We have tested all the common ones you can think of using our EFC technology, the platform with EFC technology. Just to name a few, we have the Jurkats, we have the U2S cells, CHOs, HEC293 cells, and plenty more cell backgrounds that we have run. The EFC technology is compatible. We don't see any cell background associated effect with having EFC assays reliably developed. Definitely, we do sometimes select specific cell backgrounds that works better for that assay format. But yes, we have tested many different cell backgrounds, and we are able to build very successful assays with those. And we have not seen specific variability depending on the cell background. But definitely, if you have something specific in mind, we can discuss that. And we have, as I said, we have the custom assay team. We can try develop assays in different backgrounds, see if that is something specific to a pathway. But typically, we haven't seen any specific variability associated with a particular cell background.
All right, Gaurav, thank you very much for an outstanding discussion. It seems that we are all out of time for questions today. Thank you for a great presentation. All right, if everyone can please take a moment to answer the feedback form that will appear on the bottom of your screen at the conclusion of today's webcast, we very much appreciate it. On behalf of X Economy, please have a productive remainder of your day.